Great. Yeah, thank you so much. So I'm an assistant professor of computer science and engineering at UConn. And while I'm giving the presentation, the bulk of the work was done by two amazing PhD students, Marjan Hosseini and Aaron Palmer, who were co-first authors. So most people, when they think about DNA structure, they think about the right-handed double helix, which I have a cartoon over there, discovered by Watson, Crick, and colleagues. But DNA can actually form different structures. And different structures, here's one example, in the presence of specific DNA sequence motifs. So this is a cruciform that forms under the presence of an inverted repeat. So you have these two, on each strand, you have the DNA aligning to itself. You can also have G quadruplexes. Sometimes these are called tetraplexes or G4s, and these form sheets of guanines. And G4s in particular have been associated with a lot of biological processes I'll cover in the next slide. And there's several of these. So here I'm showing all of the non-B structures that have well-known and well-characterized motifs from the non-B database. So the first thing we can know about non-B motifs is that they're conserved across species. So this hints at some functional relevance. But there's actually been some recent studies, and actually some quite old studies as well, showing that non-B DNA structures play critical roles in fundamental biological processes. So here, I'm just pointing out a few: gene regulation, telomere stability, centromeric chromatin, and increased mutability, DNA repair, inhibition, and oncogenesis. Now, G4s in particular have a lot of research associated with them. This is with respect to cancer. So stabilization or induction of G4s can lead to telomere maintenance problems, reduced oncogene expression, and genome instability. So there's two main routes in predicting where these are in a genome. So the first route is mainly experimental. And so here on the right-hand side is an example of where they looked at in the mouse genome, they were trying to look for single-stranded DNA. And so they'd used permanganate S1 nuclease footprinting to find where single-stranded DNA was on the genome. Of course, single-stranded DNA can happen just with regular transcription. So what they did is they overlaid those hits with non-B motifs. And the idea is this provides somewhat strong evidence for there being a non-B structure there. So this was done in the mouse genome, so that's a caveat. But they found around 3 to 9% of the non-B motif regions having single-stranded DNA. So take that for what it's worth, likely forming some non-B structure. Computational approaches almost exclusively use the motifs, the non-B motifs. Here's an example of the G quadruplex motif where you have this stem loop structure, where the stems are typically some run of guanines. Now, the problem, you can sort of see this in the experimental data, but the problem with using just the motif is a necessary, probably, but insufficient condition for finding non-B structures. Okay, so, and you can see that from the experimental results. These are all, in non-B windows, but a very, very small percentage of them actually had single-stranded DNA. So they're necessary, but insufficient. There was a landmark study in 2018. Oh, geez. Guiblet, I apologize for pronouncing that wrong in France, assuming that's a French last name. In 2018, where they actually associated polymerase speed in PacBio sequencing with non-B motif windows. So the idea is if polymerase is slowing down, maybe this has something to do with there being an unusual structure in that location. So this is just a cartoon of PacBio sequencing. And so you have your peaks, which you use to call nucleotides. And then you just measure the distance or the time between these peaks. And that's called the interpulse duration, or IPD. So then you can take windows that have, motif windows of particular non-B DNAs, and you can compare those to B DNA windows. So windows that don't have any non-B motif. And this is a statistic called interval-wise test, where essentially the deeper of the red color means significantly higher IPDs compared to a BDNA window, and deeper the blue is significantly lower IPDs than the BDNA window. And you can see, just taking G4s, for instance, G4s seem to have elevated IPDs, so longer distances between those pulses on the strand that has the G quadruplex, and lower IPDs on the other strand. So there are two main questions that we had with this work. First, is there a similar signature to IPDs in Oxford nanopore sequencing? So we can we extract some feature from the raw data that will be similar to an IPD that we can use to locate non-B structures? And going a little bit further from this previous study, can we actually build a computational model to find putative locations of IBD structures? Okay. So just a very high-level overview of nanopore if you haven't seen this. So this is an illustration of a nanopore. We have DNA approaching the pore. Single strand goes through the pore. It's unwound by this unwinding enzyme, and then it's ratcheted through the pore. So it's going to pass through the pore, and there's a charge running through the pore, and every so often, at regular frequency, we're going to take note of what that charge, we're going to record what that charge is. Okay? So, and that's how you do base calling, so how the charge changes as the DNA is ratcheted through this pore. Now, some of the more astute listeners may be kind of seeing where we're going here. So if there is a non-B structure upstream of this pore, right, then maybe something is happening. So maybe it's taking longer to unwind, or maybe it's taking less long to unwind. But that's what we're going for. We're actually going to say, well, there's a structure upstream, then this is going to affect how long it takes the DNA to go through the pore. So this is a cartoon of this base calling and squiggling process. So in nanopore, we're going to try to assign these observations of the charge to particular bases. And I'm cheating a little bit here because it's actually k-mers, but just for ease of exposition, I'm going to just put the base there. You can think about this as the time it takes for the base to, say, leave the pore. So top is your squiggle in base call. And then what we do is, since this is a regular interval in which we're taking note or recording this charge, we're just going to count how many specific charge observations were taken for each base, multiply that by a frequency, and then we get a translocation time. So in the bottom plot, this is what we're after for every base, a time that it takes to leave the pore. Here's what this looks in actual experimental data. So this is NA12878 and a nanopore, Oxford nanopore sequencing data set. So we have our squiggling assignment of charge to base, and then we have our conversion to translocation times. So to build a predictive model, there was quite a few struggles. The first struggle is we're a little bit weird. There's nobody, at least we couldn't find many other people that were interested in translocation times. So we had to go from the raw nanopore files and figure out how we can extract translocation times from this. To this, we had to go backwards and use an older base caller, albacore. If you were doing this nowadays, I think there are better options, but at the time, we used albacore. It's a little bit old, so we improved the base call quality by using Tombow. So this will resquiggle for us, and then we extracted our translocation times from the Tombow resquiggled files. And most of the results I'm going to show, besides the data analysis results where we wanted a longer context, we're going to look at 50 base pair windows centered around a non-B motif. We're going to look at both strands and only non-B windows that had at least 5 reads. We're going to take the median translocation time because they're quite noisy, actually. The second challenge is translocation time data is quite high dimensional. So 100 dimensional or 200 dimensional, depending on what we're looking at. We briefly tried autoregressive models, but there wasn't too much autocorrelation. So we stuck with a deep learning model instead, basically to embed this high dimensional data to a lower dimensional space where it's a little bit easy to reason or to work with. So if it's not obvious, we're going to be using autoencoders for that, but a very specific type of autoencoder. Last set of challenges, we talked about this before, non-B DNA is the label is noisy. So it's necessary, but not sufficient. So there could be a lot of motif windows where structure just doesn't form. And we don't have a ground truth data set. So a supervised approach seems out of the window. So what we do instead is we formulate this as a positive unlabeled learning problem, and specifically a novelty detection problem. I'll show some examples of what novelty detection really means and how that differentiates from, say, just regular anomaly detection. And also, because we don't have a ground truth, we're going to do something that's very similar to what you do in cisUTL analysis, where you have a lot of hypotheses. We're going to do a large-scale hypothesis testing framework, and then we're going to correct for multiple comparisons using Benjamini-Hochberg. Okay, so I just wanted to give you some intuition on how this model is actually trained. So M is the batch size. There's really three components of this model. And I'm highlighting the first component here in red and blue. So X is a bDNA sample, and then F is the encoder, and G is the decoder. So that first term is just computing a distance between bDNA sample and its reconstruction. Okay, so we want to reconstruct bDNA well. The second part of the loss function, this purple part, is a test statistic. And so we're going to optimize a test statistic. And you're probably saying, well, why not just use any sort of encoder to embed? Well, we actually want to ensure that this latent space is indistinguishable from multivariate normal. And we do that very specifically to satisfy some regularity conditions. So we can do hypothesis testing. Okay, so that's all on the bDNA. So the non-bDNA comes in, and the non-B motifs come in the second part. So in the second part, we have this max of 0 and then some parameter delta minus the distance between Zi, a non-B sample, and its reconstruction. Okay, so if this is an actual bDNA structure that's labeled non-B, then this distance is going to be very low, and this cost is going to be very high. If this is a non-B labeled sample that also has non-B structure, then that distance is going to be very high, and that's going to be lower, so better. Effectively, what's going on here, showing this sort of cartoon on the bottom, we have three sets of points. We have the A points, the black points, which are bDNA samples that are labeled bDNA. We want to be really good at embedding those and reconstructing those. So those embed well to multivariate normal, and they stay on the data manifold. We have C, which are the red points that are labeled non-bDNA, but are actually bDNA. Okay, those similarly will embed well and stay on the manifold. And then this discriminative loss term on the right, this will actually push the B points, which are non-B labeled and non-B structure, off the manifold into the tails of the embedding. So I didn't say it, but this is our method's called the goodness of fit autoencoder with discriminative loss for novelty detection. So that's where all the components come in the acronym. So finally, to make calls, we test whether or not the embedding and the reconstruction of the non-B samples follows the same distribution as the embedding and the reconstruction of the bDNA. For the embedding, we use a Mahalanobis distance from an empirical normal and L1 reconstruction for D. And then we apply BH to control FDR. And alpha level, I think all the results are 0.2. So first of all, did we find a signal in translocation times? Well, spoiler, I probably wouldn't be up here if we did not. So we did. It isn't for all non-Bs. So some non-Bs have a stronger signal. In particular, short tandem repeats and G quadruplexes have the strongest signal that seems to be preserved across quantiles. So the different colors are quantiles. The x-axis is the position in the non-B window. We're just stacking all the non-Bs on top of each other. And the y-axis is the translocation time. So zDNA and A-phase repeats, they look like they have a strong signal, but you can kind of see it's not preserved across the quantiles. And all methods found it much harder to actually find non-B structures, with the caveat that we haven't actually chemically validated these in zDNA and A-phase repeats. And then something's going on with mirrored, direct, and inverted repeats. We can talk about that offline, but we have some hypotheses why there isn't so much structure showing there. Sorry, I should have pointed this out, but control is the bDNA that's dashed, and non-B is the solid line. And this is trained on NA12878. So then we did the same interval-wise testing. Again, so the deeper the red is going to be higher translocation times in the non-B DNA motif window than bDNA, and blue is going to be lower translocation times. And this is sort of like the quantitative analog of the previous slide. So we see the G quadruplexes. On the G quadruplex strand, IPDs are much higher, okay? And on the other strand, they're much lower. You can see the same behavior in our IWT. So on the forward strand, they're much higher, and on the reverse strand, they're much lower. And this is also shown in IPDs in the paper on IPDs as well. Same trend. So then we looked at simulated data. We chose the two strongest signals, and then we built functional forms, introduced some noise. And so here are the quantiles of the simulations. And now that we have the ground truth, we can actually evaluate F1 score accuracy. So on the y-axis here is just F1 score. On the x-axis are non-B ratios. So this is the proportion of the non-B labeled samples that are actually non-B structured, so simulated from one of these models. And you can see that, I guess there's two main takeaways. Novelty detectors work better on average than your classifiers, and that's to be expected because the label is very noisy. And also, our method GoFei DND works really, really well. And I think that's due to this manifold assumption that we make very strongly. We also looked at the experimental data and just saw how many non-B structures did you call at a specific FDR control level. So this is alpha equals 0.2. We didn't bold them in the paper because it's not necessarily the higher the better, because these are not, again, validated structures, but you do want your model to make a lot of calls to follow up on. And our model does make the most calls with all non-Bs besides G4s, where one class SVM has a little bit more calls than we do. Okay, so this is, I think, a very interesting visualization with the simulated data. So remember what we're doing. We're embedding bDNA, and we're trying to reconstruct it well. And then things that don't embed well and don't reconstruct well are probably non-bDNA structures. So the light blue here is the null hypothesis. These are bDNA samples that we embed and we reconstruct. On the x-axis, we have Mahlanobis distance. On the y-axis, we have reconstruction error. Okay, so our two test statistics. And then these are G quadruplexes just with different non-B ratios. So you can see that in embedded space, the null distribution is this nice multivariate normal looking distribution. The lighter tealish color, and sorry if this is a little bit hard to see, are non-rejected DNA with the motif. Okay, so these were labeled non-B, but we didn't reject them, so they're likely B DNA. Okay, and they nicely overlap with our null distribution. So the red triangles here, and I'm highlighting them because they're hard to see, are rejected bDNA with the motif. Okay, so what does this mean? We took a bunch of bDNA and we passed it through the model, and then we saw where it embedded. Okay, now these are outside of the null distribution pretty much without any sort of structure. They're just all around. Okay, so we would call these anomalies or outliers. You know, these are just regular outliers. They're just different from bDNA. But the dark blue are different in a very specific way, right? They're biased towards higher reconstruction error and higher Mahlanobis distance. So again, this is not proof that we're finding non-B structures, but I think this is, again, some more good evidence that we're finding some signal. That's interesting. Okay, just to conclude, we did find that translocation times are associated at least with non-B sequence motifs. We formulated this problem as semi-supervised learning with a PU learning formulation. Seemed to work better. And then we have this large-scale hypothesis testing framework that enabled us to do this without any ground truth. Some caveats. We know there are other, well, at least we suspect there are other forms of translocation time variation. Methylation is one. We didn't include that in the study. We do need to do biochemical validation of these putative calls. And once you have biochemical validation, maybe a supervised learning method is preferable to semi-supervised. So this is my lab from left to right, the two co-first authors, Marjan and Aaron, and then a new incoming PhD student, Bill Monka. So thanks to the funders, data providers, and you for your attention.
Thank you for the talk. Any questions?
Thanks for the talk. I'm wondering how biochemically stable these alternative structures are. So at the point you're sequencing, there's been a library prep, there may have been a phenol extraction or a pull down of some kind. I remember quadruplexes being associated with metal ion binding or something like that. So how, if you're able to classify them with 100% accuracy in the sequencing, would that be the same you would see in a nucleus in a cell?
Yeah, that's a very, very good question. So I guess two things. We did include some biochemical analysis of G quadruplexes, particularly because there's a method that actually estimates how stable they are. Quadrant it's called. And so all methods did pretty good at calling weak and strong stabilizers based on quadrant. We weren't any special on that. And the other, for the other part, it seems like cations do stabilize many of these non-B structures. And nanopore sequencing is more preferable than other sequencing platforms, actually, because the inclusion of cations is used to stabilize DNA. So that's not a great answer, but at least, yeah, there is some evidence that it nanopore in particular can, yeah.
Thanks for an interesting presentation.
Sure.
I was wondering, do you have any insights what causes the difference in the translocation times for the non-BNA structures? Is it the interaction between the unwinding enzyme and the DNA?
Yeah, that's a very good question. So, I mean, you do still have to unwind bDNA. I think it's most clear with G quadruplexes, both from like intuition standpoint, and also we have some empirical evidence. So with G quadruplexes, you have this weird sheet-like structure which has to unwind on the G quadruplex strand. So translocation times are higher. On the other strand, it's sort of already unwound. So that goes by much faster, and it's sort of what we would expect, and it's what we find. The other, we're working with collaborators that are working with a genome where they know they have a lot of G quadruplexes. It's a very G quadruplex rich organism. And when they do nanopore, they find it breaks, like the strands break. And their hypothesis, again, this has to be confirmed, but their hypothesis is that when you have this structure, it's harder to pass through the pore, and sometimes it just breaks. So it seems to be something there, and I think that's why it's affecting translocation times.
Yeah, great talk.
Thank you.
What's your why are there different quantiles? You know, is this just nucleotide composition or something? And you see the same signal across the different quantiles.
Yeah, good question. So the quantile plots, when you're looking at things, so these are motif windows, and most of those motif windows are probably bDNA. So when we're visualizing this, well, actually the 2018 study did this as well. It's a great way to visualize it, because at the quantiles of translocation time, you'd expect that most of that signal is coming from non-B structure if it exists, because you're focusing on the translocation times that are the highest or the lowest. Now, you should take this with a grain of salt, because here we could be including outliers. It just could be methylation that's coming into these quantiles. But and that's why, especially with A-phase repeat and zDNA, where it's not preserved across the quantiles, we think a lot of that is noise. There might be structure there, but a lot of it's probably noise.